Welcome to the deep dive. Today we're getting into some really fascinating AI research. It's got big implications for anyone building or using protein language models.
Yeah, it's a bit of a head scratcher, actually.
Definitely. Picture this. Even super sophisticated models, the ones trained on mountains of biological data, they can get tripped up by simple repeating patterns and sequences.
Right. They end up showing really high confidence, these anomalous scores, where biologically it just doesn't make sense.
So the paper we're diving into is in-context learning can distort the relationship between sequence likelihoods and biological fitness by Cantrou and colleagues.
That's the one. Comes out of Yale and some other top places. And it really challenges how we think about the reliability of these models, especially for things like predicting fitness or designing new sequences.
Exactly. Those likelihood scores we often rely on, they might be a bit shaky under certain conditions.
So for all you AI researchers, you ML engineers working with biological sequences, this is definitely need-to-know stuff. The core issue they raise is that sequence repetition can mess with how well the model's likelihood score actually reflects biological plausibility.
And it's not just some theoretical edge case, right? This could genuinely affect real-world results.
Absolutely. So, okay, let's do a quick summary for that first minute. Basically, in-context learning, especially in transformer models, the ones we use a lot for proteins, can twist the connection between sequence likelihood and actual biological fitness.
How?
Well, when you have repeated motifs in a sequence, the models tend to give them these artificially high likelihood scores. You see it as really low pseudo-proplexity.
Okay.
And it seems to happen because the model is basically doing a lookup trick within the sequence itself.
Ah, like finding the answer key in the input.
Pretty much.
Okay.
And this retrieval thing can actually override the model's deeper learned knowledge about what makes a sequence biologically sound.
Wow. Does it have to be perfect repeats?
No, that's another key finding. It extends to imperfect repeats, too. And for larger models, even things like reversed complement motifs in RNA, which are biologically important.
And there's an impact on embeddings, too.
Yes, exactly. Repetition can also degrade the quality of the sequence embeddings the models produce, which isn't great for downstream tasks.
Okay, so our mission today is to really unpack how this distortion works. We'll look at the mechanisms, the differences between model types, transformers, convolutional, others, and what it means for protein and RNA sequences.
Right. We want to equip you with a clearer picture of these potential pitfalls when you're interpreting what these language models are telling you.
All right, let's dig in. Where did the researchers start?
So it kicked off with an observation. They were looking at a whole bunch of protein domains, evaluating them with ESM2, specifically calculating pseudo-proplexity.
And pseudo-proplexity, just as a quick refresher.
It's basically how uncertain the model is when it tries to predict a masked part of a sequence. So low pseudo-proplexity means the model is very certain, very confident, it assigns a high likelihood. Got it. Low score, high confidence.
Exactly. And they noticed something interesting when they looked at the sequences that had these incredibly low scores, like really close to 1, near perfect certainty. You can see this distribution in figure 1 of the paper.
Okay, and what was the pattern?
A lot of these super confident sequences had obvious repeated motifs in them. Figure 1 actually shows a few examples, makes it really clear visually.
So it was necessarily deep biological insight driving the high scores.
That was their hypothesis. They suspected the models weren't grasping some profound biological principle, but were instead, exploiting the repetition.
The lookup mechanism you mentioned earlier.
Precisely. The idea is, if part of a repeated section is masked, the model just looks at the other copy of that same section within the input sequence to figure out the missing piece. That's in-context learning in action.
Like it builds a temporary cheat sheet just for that sequence.
Yeah, that's a good way to put it. It's learning on the fly from the input itself, not changing its fundamental learned parameters. Okay, that makes sense. So how did they test this lookup idea more rigorously?
They did something pretty simple, but quite clever. They just took protein domains and doubled them. Literally stuck a copy onto the end of the original.
Just duplicated the sequence, and what happened?
With the transformer models, like ESM2, they tested both a big 650M parameter 1 and a smaller 8M version. They saw this dramatic collapse in uncertainty. The pseudo-proplexity just tanked, went down to nearly 1.
Wow.
Figure 2a shows this really clearly. It's a massive drop, suggesting the model suddenly became incredibly sure about these doubled sequences.
Which is weird, right? Because just doubling a sequence doesn't automatically make it better biologically.
Exactly. It strongly points away from genuine biological understanding and towards some kind of pattern matching.
Did other types of models show the same thing, like autoregressive ones?
Good question. They tested Progen 2M, which is autoregressive. It did show a significant drop in perplexity for the doubled sequences, but not that complete collapse like ESM2.
Why the difference?
Well, autoregressive models only look backwards at the preceding context. They don't have that bidirectional view that masked language models like ESM2 do, so they can't see the upcoming repeat quite as easily to use it for prediction.
Ah, so the bidirectional nature of transformers seems key here.
It seems to be a big factor. But the real clincher, the thing that really suggested it's about patterns, not biology, was when they tried doubling random sequences.
Sequences with no biological meaning at all.
Exactly, just random strings of amino acids. And guess what? Doubling those also caused the uncertainty to collapse in ESM2. Figure 2b shows this. Progen M also showed a decrease.
Okay, that's pretty convincing evidence. It's leveraging the repetition itself, regardless of content.
It really looks that way.
So transformers seem prone to this. What about other architectures? Did they look at convolutional models?
They did. They tested CRRP, a big convolutional model, and its behavior, shown in Figure 2c, was different again. CRP only showed that uncertainty collapse for repeating units that were relatively short, less than about 70 amino acids. For longer repeats, the uncertainty decreased more gradually as they added more repetitions.
Interesting. So it's like CRP has a shorter attention span or memory for these repeats.
That's kind of how they interpret it, yeah. A sort of limited operational memory compared to the transformers, at least for recognizing and using these longer patterns.
So it's not a universal issue across all architectures. What about the newer long context models, things like Mamba-based ones?
Yep, they looked at LCPLM, which is based on Mamba-S. And interestingly, as you can see in Figure 2D, it didn't show a complete uncertainty collapse, even with short, highly repetitive random sequences.
Really? What did it do?
It showed a more gradual decrease in pseudo-proplexity as the number of repetitions increased. This suggests these newer architectures might handle long-range dependencies and repeats differently than standard transformers.
Okay, so to recap, repeats lead to low pseudo-proplexity, especially in transformers, likely due to a lookup mechanism confirmed by the random sequence doubling. But different architectures react differently.
That's the gist of it so far.
So how did they nail down this idea that the in-context lookup is actually overriding the model's learned biological knowledge?
Right. This is where Figure 3 becomes really important. They used a specific example, a Calmodulin binding motif.
Okay.
When they masked a residue in just one copy of this motif, ESM2 correctly showed a preference for hydrophobic amino acids like tryptophan W, which is biologically correct for that spot. That's Figure 3a.
Standard stuff.
Standard stuff. But then they added a second identical copy of the motif to the sequence. Now, when they masked that same spot in the first copy, the model became absolutely certain it had to be tryptophan. The confidence shot way up. That's Figure 3b.
So the presence of the second copy amplified the confidence.
Massively.
Yeah.
But here's the clever part. If it's a lookup, masking the same position in the second copy should break the lookup right.
Yeah, it removes the answer key.
Exactly. And that's what happened. Figure 3c shows that when they masked the equivalent residue in the second copy, the uncertainty about the first position came back. But if they masked a different non-equivalent position in the second copy, Figure 3d, the model stayed super confident about the first position.
Because the lookup source was still available?
Precisely. It's strong evidence for direct retrieval. They even scaled this up across thousands of domains using entropy to measure uncertainty, and the pattern held, as shown in Figure 3e. Masking the equivalent position consistently restored uncertainty.
It really behaves like it's just copying from the other motif.
It does. They even compared it to how induction heads might work in autoregressive models, though the mechanics in bidirectional models might differ. But here's the kicker.
Go on.
What if the information it retrieves from the second copy conflicts with what the model has learned biologically?
Ooh, a clash between the lookup and the learned rule.
Exactly. They took that Calmodulin motif again. Remember, the model preferred tryptophan, W, at that key spot.
Right, the hydrophobic one.
So in the second copy, they deliberately changed that tryptophan to a lysine K, which is positively charged. Very different.
Right.
Then they masked the original position in the first copy. What did the model predict?
I'm guessing lysine.
Lysine, with high confidence. That's Figure 3F. The in-context information The K it saw in the second copy completely overrode its learned preference for W at that position.
Wow, that's quite something. It ignores its training just based on the repetition.
It really highlights the power of this in-context retrieval. And figure 3i shows this generally holds true across many proteins and random sequences. The model's prediction heavily leans towards whatever residue is at the equivalent spot in the repeat.
You mentioned an exception for tryptophan and cysteine.
Yeah, slight caveat. For W and C in natural proteins, the effect was a bit weaker. The thinking is maybe because they're rarer amino acids, the learned prior for them is stronger and offers a little more resistance to being overridden.
Still, the overall trend is clear. The lookup often wins.
Definitely. They also did some experiments introducing small insertions to create ambiguity about where exactly to retrieve from. Found this interesting contralateral preference. It tended to look right if the mask was left and vice versa, maybe favoring the longer uninterrupted repeat section. Figures 3G, 3H, and 3J show that.
So this lookup isn't just for perfect identical copies then. What about the imperfect repeats we see in biology?
Great question. And no, it's not just perfect repeats. Figure 4a shows how they tested this by creating sequences with mutations, substitutions, insertions, deletions in the repeated copy.
Okay, making them similar but not identical.
Right. And figure 4b shows that even with significant differences, the pseudo-proplexity of the mutated part was still lower when it was paired with the original copy.
So the model can still retrieve information even if the match isn't exact?
It seems so, yes.
Yeah.
The retrieval mechanism is surprisingly robust to variations.
And they did that needle in a haystack test too.
Yeah, that was neat. They put two identical short random sequences, the needles, inside a long stretch of unrelated random sequence, the haystack. That's in figure 4c.
And could the model find one needle using the other?
Yes. Figure 4d shows ESM2 could do this reliably for needles as short as 10 residues, even when separated by hundreds of unrelated residues in the haystack.
But there was a limit.
There seemed to be, yeah. An operational memory limit around 1,000 residues. If the needles were further apart than that, the retrieval ability dropped off.
So it can find patterns across long distances, but not infinitely long. What about that one skip experiment? That sounded intriguing.
Right. That suggests something even more complex might be happening. These sequences, shown in figure 4e, had matching residues only at every other position, like ACGT.
So no adjacent matching pairs.
Exactly. But the model still behaved as if there were repeats. It assigned high probability to the residues at the corresponding skipped positions, figure 4f. It suggests the model might be doing something like an internal sequence alignment, figuring out the correspondence even without a continuous block of identical residues.
That's quite sophisticated pattern matching, almost scary.
It's definitely impressive pattern recognition. But, as they point out, what patterns the model learns to recognize like this depends heavily on the training data. Which brings us to the RNA experiments.
Yes, moving beyond proteins.
They used RNA language models, RiNALMO, and tested different kinds of appended context. Not just repeats, but also complements, reversed repeats, and reversed complements. Those reversed complements are important because they form hairpin structures in RNA. Figure 4G outlines this setup.
And what happened? Did repeats cause the same collapse?
Yep, repeats cause the uncertainty collapse in both the small and large RiNALMO models, just like with proteins. That's figure 4H.
Okay, expected. What about the others? Complements, reverse complements.
This is the really insightful part. Neither model reacted much to just reverse sequences or just complemented sequences. But the larger RiNALMO model, the 650M parameter 1, did show uncertainty collapse for the reversed complements, the hairpins.
But the smaller one didn't.
Correct. Only the large one reacted strongly to the biologically relevant hairpin structure.
What does that imply?
It suggests the larger model learned the reversed complement, or hairpin pattern, as a single composite unit, likely because those structures are common and statistically enriched in the RNA training data. It wasn't just combining reversed and complement.
So the in-context learning is sensitive to the statistics of what it was trained on. It learns common biological motifs non-decomposably.
Precisely. It's not just abstract pattern matching. It's pattern matching shaped by the biology and the training data.
Fascinating. Okay, so we've seen how repetition messes with likelihood scores. What about the embeddings? You mentioned those earlier.
Right. The embeddings, those numerical representations of sequences, are super important for downstream tasks. So they investigated if repetition affects their quality.
How did they measure embedding quality?
They used a task called one-fell-swoop regression, shown in figure 5a. Basically, you use the embeddings to predict masked parts of the sequence. Better embeddings should lead to better predictions, meaning lower error or loss,
And they compared embeddings from sequences with lots of repeats.
against embeddings from control sequences of the same length, but with only one copy of the protein domain and the rest filled with random sequence.
And the results.
The embeddings from the repeated sequences performed worse. They had a higher validation loss on the regression task, as you can see in figures 5b and 5c.
Worse than the single copy plus random padding.
Yes. And the performance generally got worse as the number of repetitions increased.
So repetition doesn't just inflate likelihoods, it actually seems to degrade the information content in the embeddings themselves.
That's what the results suggest. It seems adding repetitive content is more detrimental to the embedding quality than just adding random padding.
Okay, that's a lot to take in. Let's try and summarize the key takeaways from this deep dive, especially for the technical folks listening.
Sure. First, and maybe most importantly, be really careful interpreting likelihood scores from these models, especially protein LMs if your sequences have repeats. High scores might just be that lookup artifact, not real biological plausibility.
Got it. Caution with likelihoods and repeats.
Second, transformer architectures seem particularly prone to this in-context retrieval, causing uncertainty collapse.
Okay, transformers are susceptible.
Third, other architectures like convolutional or state space models react differently. They have varying sensitivities, which might point towards ways to build more robust models down the line.
Architecture matters.
Fourth, this in-context learning is pretty sophisticated. It goes beyond perfect repeats, involving complex pattern matching, maybe even something like internal alignment.
It's a powerful pattern finder.
Fifth, what patterns it learns to exploit like this is heavily influenced by the training data statistics. The RNA hairpin example really drives that home.
Training data shapes the shortcuts.
And finally, sixth, remember the embeddings. Repetition can actually hurt the quality of those sequence representations, potentially impacting downstream tasks.
Embeddings can degrade. Okay, those are crucial points. So, thinking bigger picture, what are the implications here? Sounds like a potential weak spot for sequence models in biology.
It definitely highlights a potential failure mode, yeah. Especially since repeat domains are pretty common in nature. It means we need to be aware of this when we use models for designing sequences or predicting fitness.
So future work needs to focus on mitigation.
Absolutely. Finding ways to lessen these distortions, whether through changes in model architecture or different training strategies. It also makes you wonder, does this happen with other types of biological data, like models trained on 3D structures? Do they have similar in-context retrieval issues?
Good question.
And overall, it's just a solid reminder that a really low perplexity score doesn't automatically mean the model has a deep understanding of the biology. It might just be very good at spotting superficial patterns.
Efficiency in pattern matching isn't the same as biological insight, which leads to a final thought, maybe a bit provocative.
Let's hear it.
Could these shortcuts the models take with repetitive sequences, could they, in some weird, abstract way, mirror how biology itself handles redundancy, or how it exploits repetitive motifs for function? Obviously, biology does it in a much more complex, meaningful way.
That's interesting. Like, is there a distant echo? Our models find a simple pattern matching trick, while biology uses repetition for maybe structural stability or binding interfaces or evolutionary modularity.
Exactly. It's just a thought, but this study really sparks a fascinating conversation, doesn't it, between the biases baked into our AI and the actual grammar of life.
It really does. Makes you think about what these models are really learning.
Well, this has been a fantastic deep dive. Thanks for walking us through this important paper.
My pleasure. Lots to consider.